Today we're going to talk about our project. We're going to talk about how beekeepers can look for and find Varroa-resistant traits to understand the biology behind it and how to monitor for it. I just wanted to thank, we've had some fantastic mentors here, some of which I'm sure you'll know, in Ralph Buchler and Stephen Martin, who helped us with the start of the project and some tricky questions through it. We also spent quite a lot of time trying to understand the work that the USDA was doing, which is a quarter of a century ago now, but still reads pretty well. And also to thank Tom Seeley for his encouragement and ideas, and particularly the learnings from the Arnott Forest. This is us. We're only half an hour away. There's nothing particularly unusual about our hives or our style of beekeeping. It's a pretty typical area for beekeeping. It's pretty similar to probably most of yours. We have about 130 colonies now in the project across just over 30 apiaries. And what we've been doing is to follow the research on how to select for the traits that you're seeing naturally in colonies which are surviving. Nothing complicated. So we're going to talk through what is Varroa resistance and some background to it, and increasing amount of education that's available. Why selecting for these traits is really important. It isn't just about getting away from chemical treatments. It's more fundamental than that. How we define Varroa resistance, I think there's a lot of flaky terms gets thrown around in this area. And when it's your bees at risk, you better try and get it right and understand what you're doing. And then we'll take you through some of our own case studies. So we've done quite a bit of our own research because we didn't think we could take decisions on our colonies without really understanding why. And I think having research papers waved at you isn't good enough. And then we'll do some wrapping up. So you may have seen this picture before. It's from a research report from Barbara Locke. And it was, at the time, it was where the main resistant colonies were. So out in Primorsky in eastern Russia, which Jacques talked about yesterday, we've now got most of South Africa is Varroa resistant, and most of South America as well, right up into Central America. And I've added Cuba on as well, which is the largest area or largest island of resistance, over 200,000 colonies there. And so it's quite a remarkable story. We've got big patches in Europe of mite susceptible bees, including the UK and also through North America. So we just need to understand this. Why has it taken only 5 to 8 years in places like Cuba and South Africa, and why we've still got a problem over 30 years later? This is back to the UK. So these are case studies from the website that Stephen Martin led on, the Varroa-resistant.uk. It's a free-to-use education/science website, and the case studies there, if we just march around the country quickly, at Lund Valley is over 70 colonies. The whole club doesn't treat. In northwest Wales here, you've got the biggest area of Varroa resistance. In the UK, they've just celebrated their 16th anniversary of not treating. There's 500 colonies involved there. We've got a couple of commercial beekeepers with the Foragers Bee and Honey Company and fen apiaries of 60 to 80 colonies and about 100 at fen apiaries. And then there's us down in the bottom corner here. And just an interesting point is that all of these colonies are open mated. All the queens are open mated. You often hear that instrumental insemination is necessary, and it's not. If we add on to that map all of the beekeepers which hadn't treated for at least 8 years, this is from a couple of years ago, you can see the Varroa-resistant traits are all over the UK. They're pretty much everywhere, and this is particularly where there are locally adapted bees, where's that continuity has taken place. So they'll be in your hives as well, so it's just a question of identifying them. Education in this area is improving rapidly. I think we should give credit to the late Ron Hoskins for the work that he did. Was well ahead of everyone else. There's been some good early articles in the BBKA magazine and also at the Spring Convention, good airtime there. The BIBA conference 2018, I think, got a lot of people interested. And then the National Honey Show here. There's been a publication, a good one. I think it's only £5.50 from the BBKA. It's probably available downstairs. And it's become a topic for the NDB. So it's been tutored, I think that's right. It's been tutored the last few years, which is good. So there may be a trickle-down effect. We'd love to see it in mainstream education. I think it's probably about right time. There's been some apiary workshops, which we run, but also the early ones were run by the Hudsons up in northwest Wales. They've had international delegations come through from places like Switzerland and Germany. We've had a book published, and there was a good series of articles in Beecraft. So this is happening all the time. The National Bee Unit there, I'm afraid it's really quite a negative amount of feedback we get from them, and I don't know if it's a policy thing or whether it's people, but they really could be doing a lot more proactive research on Varroa resistance to try and find a proper solution. And the USDA has done that. So they put a lot of time and effort into it, and their website is excellent. And there's some early research here which showed they imported some bees from Primorsky, the Eastern Russian bees, the resistant ones, and it shows the estimated mite loads versus the US commercial bees. So they didn't understand the traits at the time, but this was, this is over a quarter of a century ago. So, and that proactive response from the USDA has spawned an awful lot of bee breeders in the United States, which are selling either, you call them treatment-free bees or VSH bees. We don't really have anyone doing that in the UK, so there's a great opportunity there. Right, so why selecting for Varroa resistance is important. Our current cycle is that we have really no selection for Varroa resistance in most places outside of our club. So that means you're breeding bees which are susceptible to Varroa. You need to treat them to keep them alive, which seems perfectly normal, and that's been our teaching for the last 20 years or so. And when you keep these bees alive, you then breed from them. You've got drones flying, you might be reproducing from them. So in effect, you can be keeping bees alive with low hygienic traits against Varroa. And at the same time, almost regardless of how many miticide treatments you put on those colonies, there will always be some surviving mites. And those surviving mites are tough. So they are surviving multiple amounts of oxalic acid, sublimation, perhaps a spring treatment and a winter treatment as well. So you're selecting for really tough, resilient mites at the same time as breeding from bees which don't have the right traits. So there's the effect. So we have actually been perpetuating the Varroa problem. And that's why we've still got a Varroa problem 30 years on. If you think about the balance of Varroa and honeybees working together, if you went back 25 years, one dose of Apistan would suffice. But now we've got these multiple treatments and engagements with the beekeeper. So we've become experts at how to apply these miticides. We know about temperatures, we know about time of year, we know about the PPE we might use, but we've not become experts at the interaction between Varroa and honeybees. We've sort of stepped back from that. Once you start selecting for Varroa resistance, it all changes. You then get resistant bees, they don't need treatments. You then start spreading the right traits. You've got the drones flying, you're splitting them at a time of reproductive swarms, and then you have a low level of mites, which the bees keep down. So what is Varroa resistance? Firstly, it's just step back a little bit and think this mite has come from Asia, and it's used to just breeding in seasonal drone brood. And seasonal drone brood out there is not dissimilar to what we have over here. And if mites only bred in seasonal drone brood, it would not be a problem for us at all. But when the mites came over here, they could not believe their luck that they are now able to breed in all of the worker brood. And for us, it's about January or end of December, right the way through to November. And that's been their opportunity. And that's why mite loads have been so big. So the problem was breeding in worker brood, and the solution is also the worker brood as well. So that's the bit that the bees have to get right. There's lots of different traits that get passed around and talked about. And we heard a few of them yesterday. And we see these as sort of ancillary and helpful environmental factors. So propolis. Having a propolis brood envelope around a cavity is incredibly helpful for the bees. It's useful. It's quite easy to do in your own boxes just by roughing up the boxes. And also insulation. We know from the work of Derek Mitchell and others that insulation is incredibly helpful to the bees. But if we think about what happened when Varroa came to the Arnott Forest, there was devastation amongst those bees, and those bees were in their ideal place, in the tree cavities, well insulated, lots of propolis around, so it didn't save them. So propolis insulation is really helpful. You'll hear from more natural leaning beekeepers that low intervention is key, and again it's helpful, not to stress the bees too much, but we do sort of pretty normal beekeeping. And our friends up in North West Wales exactly the same, and some of those other case studies exactly the same. So you can get away with doing normal managed beekeeping and still having Varroa-resistant bees. Brood breaks. Brood breaks are good. They're healthy for the colonies, and our bees will try to swarm once a year, but the discussion that multiple brood breaks are required is just wrong. And the simple way to think about brood breaks is that we had brood breaks before Varroa arrived, and then we had brood breaks after Varroa arrived, but still colonies died. So brood breaks are helpful, but they're not the solution. Natural selection. We're not very good at that as beekeepers. We all sort of know that it's important, and we think it should apply somewhere else. But once you stop treating and getting Varroa-resistant bees, you're effectively going to have some natural selection over winter, which will help. So these are the big four which we think about, and virus tolerance is an interesting one. Barbara Lock did some work in Sweden where she injected larvae and pupae with deformed wing virus and acute bee paralysis virus to see how long the bees would live, and she compared colonies which were from Varroa-resistant colonies with those which were not resistant, where managed colonies needed treating to keep them alive. And in each case, the resistant bees lived longer. Well, that's interesting. They then copied that assay in the Netherlands, in Norway, and in France. And in each case, the local resistant bees lived longer than the local managed bees. So there's something about virus tolerance. We don't fully understand it at the moment, so more research required. I don't think it's something that's going to trouble us beekeepers. It's the researchers need to do that for us. But it's there. It's like icing on the cake. The other interesting one is mite mutilation, and again we've got a slightly different view on that. We like others think that mutilation and grooming must be part of the cocktail of Varroa resistance, and so we started looking down microscopes at damaged Varroa and cheering every one that we saw. I mean, it was just fantastic. Bits of carapace missing, antennae, legs hanging off. And we thought, this is great, isn't it? It just sort of confirmed what we were reading. But here was our problem. So this hive 5 here had about a quarter of its mites, which were falling down to a Varroa board underneath an open mesh floor. About a quarter of them were all damaged. What we couldn't understand is why the mite count was so high. So over 3,000 mites from that colony in 1 year. And that compares in the same apiary, the same setups, considerably lower numbers. So we couldn't understand this. Lots of grooming and mite mutilation, but actually the results were wrong, and something was wrong, and we made mistakes here, okay? Because we started breeding from this one, thinking it was fantastic, what a great trait it had, but the numbers didn't stack up, and it was Stephen Martin actually who helped us out subsequently, which was to explain that probably these mites were being damaged when they were being cleaned out of the cells. So the mandibles holding onto mites, cleaning them out, they'd die, they'd fall down onto the floor, we'd celebrate it, but wrongly. And subsequently, there's a bit of work from Isabel Grindrod and Stephen Martin, which seems to suggest that actually grooming and mutilation has been overrated. And part of that is based on the research which came out of Asia from Peng in 1987, which showed That's where they claimed that Apis cerana had this ability of grooming. But the experiment actually took mites which were being produced in an Apis mellifera colony. And what happens with mites is that they're pretty smart. They take on a chemical camouflage. So they take on the smell of the host. So when you put Apis mellifera mites in a colony of Apis cerana, it's not surprising they get a bit itchy. Right? So it was probably an exaggerated outcome, and that bit of research got cited all over the place. So we started to, when Varroa came over here, we got quite excited about that, and it seemed to be fixed, that mutilation and grooming was important. We don't think it is, and we don't even select for it anymore. So that leaves uncapping and recapping, and chewing out of infected pupae. So let's have a look at uncapping. So uncapping is helped by the microporous cell cappings. Now this is using electron microscope. That is a little hole in worker brood, and then you can see some hairs there on the other side, which is bits of the cocoon which is stuck to the top of the cell capping. And volatile compounds are coming up through the cell cap, which enables bees to check the hygiene of what's going on. But it enables them more than that as well. So this was a bit of work done by Fanny Monday's team. And what it shows is that this is the level of proportion of uncapped cells. So for dead or damaged pupa, they would uncap over 80% of those cells. Not a surprise, bees know the difference between health and issues. But here's the interesting one. They will uncap, if you got the scent of offspring of Varroa, they uncap over 70% of those cells, and that was the big breakthrough. So they can smell Varroa offspring through those cells. And interestingly, if it's just a single female in a cell, then the uncapping is very low, just over 10%. Okay, so it's the offspring which is the key to Varroa resistance. This is thought to be in the antennal lobes. And if we had Martin here, no, he's gone, isn't he? He could probably tell us how that's comes through to the mushroom bodies. But it's for bees which have a particular ability, olfaction ability. We call them detector bees. They're about 11 days old, nurse bees. And here's a bit of a close-up. You can see this happens at the sort of purple-eyed stage, pink to purple-eyed stage, which is quite key. This is interesting. You can see the uncapping is taking place. You can see the silk cocoon underneath still, so they haven't quite broken that through, but they open it up to have further investigations. And if they're healthy, they'll recap them again. This is typically what we see in our colonies, and I think a lot of colonies, right, certainly people I know who've got resistant bees, it's sort of a cluster effect of different unopened cells. And what's happening here, there's in that cluster there might only be 1 or 2 infested cells, but the chemical compounds are coming up through the microporous cell cappings, and they're being blown around by bees, either with wings or just with bee activity, and that leads to a level of inaccuracy. So some work's been done behind that called spatial recapping, and it's and they've been able to prove that's the case. Here's just some pictures. There's almost a communal effort to get rid of this infected pupae here, again at the purple-eyed stage, interesting. And when this gets chewed out, we get to see that. So we get to see the bits which come down through the open mesh floor onto the Varroa floor underneath. So let's just see why the purple-eyed stage is important. So here's the 12 days of a larva becoming imago, and if we overlay the Varroa reproduction here, so the mother mite's going to pop in up to 2 days before, her egg laying starts about 3 days later. It's firstly a son and then a series of daughters. Now the blue line indicates when the son is mature enough to mate with his sisters, and the magenta lines are when the sisters are mature enough. So roughly it takes to about day 10 before there's any mating activity, but that's when the uncapping and chewing out takes place, okay? So it happens before the reproduction has occurred. So when you see uncapping next time at the pink to purple-eyed stage, you'll know. And if there's one slide to take away, I think it's that one there. And why is this important? Because the founder's mite has a finite reproductive capacity. So she'll mate 2 to 3 times, and that's it. And so in resistant colonies, what you find is a very large cohort of infertile mother mites. So there's an average here, it's just an average, but 45%. So often the mites that we'll see, the dark red mites we see have fallen down. If there's a lot of them, we'll think, well, just under half are probably infertile. So this is quite a simplistic diagram just to show you, in effect, what's happening. So these are the mite-susceptible bees, the ones that you can buy from the bee suppliers, and ones that we're all keeping alive, wrongly keeping alive. So the mother mite will go in and she will mate. We're just going to assume that she's successful and has 2 daughters. Those 3 will then mate, and so on. And in the end, in 3 generations, you've got a small army of Varroa, okay? And that's when you see those great big graphs of exponential growth of Varroa. That's what's happening. And that's what would have happened when Varroa first arrived. At the same time, you get a very large virus load on your bees. So these bees at the pupa stage are being injected with virus. So you get a pretty unhealthy colony. But if you look at the resistant bees, what's happening is that the mother mite here will be chewed out by the bees, and because they found there's potential for offspring there, she'll have another go, and then she becomes infertile. So for the sake of sacrifice of 3 bees, you've got a colony which has low level of mites and a low virus load. We've lost the sound, but this is Jeff Harris, who was at the USDA and was in the lab when they found VSH bees, so very sensitive hygiene bees. And it's going to show you a bit of the uncapping process and then the chewing out process. And keep an eye on the chewing out process. See the pink to purple-eyed stage here? You see the mother mite coming out? So in the chewing out process, in the chewing out process, the mother mite escapes, but all the offspring perish. Okay? Do you want to know whether that foundress mite escaped or not? Yes. She did. We lost a few seconds off the video, right? She's incredibly quick, right? And they're hairy, and they're used to living on bees, you know. And she climbed onto the back of the neck, and that's as far as we know. That's as much of the story. So tune into next episode. Okay, so that's what happens in practice, okay? And we've seen it a few times. Usually your phone is on the inside of your bee suit and you see it happening, and the time you grab it, you're wrestling with bees on the inside of your veil, and you start narrating badly. You know, it goes horribly wrong. But right, so we'll come back to recapping, okay? We talked a bit about uncapping and recapping. So the detector bees have this ability, and recapper bees, I mean, it's just their job, right? They have probably don't have the same olfactory ability, and if they see healthy pupa, they go and recap it. And there may be some pheromone involved, and scientists have got interested in this, at the uncapping level of uncapping and recapping. They've started trying to measure it. So they actually peel the top of the cell open, and they can see where there's a sort of cemented patch underneath it, where the silk cocoon has been broken. So they go around measuring these things in resistant colonies and non-resistant colonies, and they see it as a proxy for Varroa resistance. Okay, so it's a level of activity that's going on. So let's just look at the resistant bees populations over here, and there's Cuba, and it's got almost 70% of all worker brood is uncapped and recapped. Isn't that remarkable? Right? And then in England, we've got numbers here about 60%. That's from a commercial apiary, from a lady who's an expert in this area down at the front, Rona Toft. And then in Sweden, over 40%, and so on. So a lot of activity. But if you look at the susceptible population, so these are the ones being treated and need to be kept up to keep them alive, the level of activity is a lot lower. And then in naive populations, so they've never seen Varroa before, so we have some in Isle of Man, the colonies, parts of Australia still, just. So it barely happens. It happens a little bit. I mean, uncapping isn't unusual. You see it in chalk brood, wax moth, and sac brood as well. So it's something that the bees naturally do. The difference between the red and the yellow bars is that the yellow bars is where they've accidentally uncapped it and it's been recapped, where there was no infestation. So this comes back to this sort of accidental targeting, which does go on. But it also shows, because the red bar is higher, that actually they are more successful at finding the Varroa than more often than not. We don't look for recapping, okay? I think it's become a scientist's plaything, excuse me, and it's something that's interesting, but it's very hard to see in colonies. Right, we're just going to go through some case studies that we were working on. This is, again, you can see it's a pretty normal looking colony, a national. They're darkish bees. They've got darker, interestingly, over the years. They're all locally adapted bees. We just use the same bees all the time. Pretty pleasant most of the time. And they try to swarm every year, but just usually once. That's, it's pretty normal stuff. This is the whole year's analysis of that colony, which we're going to break down, showing mite drop in the yellow, hygienic behaviour correlated with worker brood along the bottom. So just looking at the worker frames of worker brood, so we've had to guess a little bit out of season, but you can see the growth through the spring there, growing into an artificial swarm. And we do try to artificially swarm them because we have neighbours and things, so there's a bit of social conscience involved. Then you see the new queen starts laying here. So the daughter of our queen, there's no new queens introduced. We try not to do that at all. And then a gradual fall away into the autumn. If we look at the mite drop over the top of that, I'll just pick out a couple of things. So you can see when the drones are being laid, there's very little hygienic behaviour against drones. So there'll be an uptick in the amount of mites at that point in time. But there's a neat offset at the time when there's a brood break after an artificial swarm. And then there's an interesting time in the summer. So the summer there, sort of July onwards, you've got a time where we're really taught that's when we should be looking at our Varroa levels to see if we're going to treat or not. But actually that's when the Varroa are moving back into the brood. They've had a brood break and they've been hanging on to the bees, and here's their chance to reproduce again. So at a time where we're looking at mite drop or doing a sample, all the mites are going to be in the brood. So you're going to get a false reading and you'll make a false decision on the back of it. This is interesting. This worried us when we first started. We didn't understand it at all. 661 mite drop. I mean, if you had that, you'd think you've done a chemical treatment, you've done a great job, wouldn't you? And it worried us because we didn't understand it, and no one's done this research before. So we've had a bit more of a think about that. So the cause of it is this fall away in the brood here. So as it comes into winter, so there's less places for mites to reproduce, and you get this multiple invasion of mites into the same cell. So there can be 2 or 3 mites going into the same cell. You also get something called stores secure, pre-winter hygienic behaviour, which a phrase we got from Torben Schiffer. And it was explained that if the bees have enough stores, they will carry on doing hygienic behaviour. And the trigger for that is when you start seeing a lot of propolis coming in, that's when the bees are getting ready for winter, and they'll be doing hygienic behaviour in the nest. The last thing you want to do is to leave them short of honey or other stores and force them into more foraging or making honey, form of honey out of sugar syrup, because they'll do less hygienic behaviour. So everything is subordinated to stores at this time of year. So just thinking about that 661 large number. So this is a typical brood frame, which we'll see this time of year. In fact, it was taken a few weeks ago. And on that, you've got about 1,100 cells of brood, okay? So across both sides, just over 2,000. And if you think about the number of frames that we had through September, October, November, there's the production of your winter bees. So you've got about 20,000 winter bees there. So should you start worrying about 661 mite drop? Probably not. And so that's why these colonies are getting through. And a number where we would have been, and most people would be very emotional about over 600 mite drop. You just have to sort of step back and think what's going on. So the next slide needs a little bit of a key. So the green P is a white exoskeleton, which is falling down underneath through the open mesh floor. And then the UN is uncapping that we've seen at times of inspections. So again, this is the full year. This is interesting in that at the start of the year, you can see chewing out of pupa going on right at the start of the year. So this is great news. You've just got rid of all of your family that's been eating out of house and home, and you're a bit itchy to do some beekeeping. And so you pull out that board, and on there is pupa exoskeleton. So what's that telling you? That the queen is laying, thank you. You've got a laying queen, fantastic, eh? So sit back 15, 16 days early, she's laying. But the other bit of good news is that already at the start of the year, your bees are taking on the Varroa. There's no hanging around here. This is, these bees have the ability to do it. And so there's 3 bits of good news there. I mean, it's a good start to the year. And in fact, here, so this is the end of December, the queen would have been laying 15 to 16 days earlier, which is why we know that the broodless period for us on this particular year was probably around the end of November rather than December. Other interesting things, so there's your artificial broodless period here. Here's the new queen that starts laying after the artificial swarm. That's the daughter of the other queen. She only really owns the nurse bees by around about August. So her daughters will have taken over the hygienic behaviour, if there's going to be any. And there it is, right? So very strong hygienic behaviour at this time of year, this pre-winter time. So there's a very strong heritability that we see from daughter to granddaughter to great-granddaughter, which is why we try to keep the same queens and the same lineage. So very heritable trait, Varroa-resistance. The other thing we see is less hygienic behaviour during the strong part of the nectar flow. And I think the reason for this is that the whole place is focused on processing nectar. It's coming in, there's pollen as well. You need to be a receiver bee, you need to process that nectar and put it upstairs with a wax cap on. It's a huge amount of work. The colony is in honey factory mode and prioritising, quite rightly, their winter stores. So there's the whole analysis again. We went a bit further because we often get the question that says, well, what's actually happening to the level of mites through the course of the year? And that's not easy without ripping the hive apart. So, but we've now got mite data, so we can have a go at that. So the start of the year, we see quite a bit of activity against mites because we can monitor that without being invasive in any way. So we think initially it's quite flat, then there's growth during the drone period because there's a lack of hygienic behaviour at that point in time against drones. And then we see a slight fall through the broodless period after the artificial swarm. Then we see this growth, which is or estimates of growth during the nectar flow, where the bees are focusing on processing nectar. And then we get this pre-winter clean out, so it falls again. So in effect, a resistant colony should have, at the end of the year, roughly the same amount of mites than it did at the start of the year. And this is how we think about it. So if you stay on the green line, so each year you get a little bobble effect of drones and then the strong summer nectar flow. and it should finish about the same. So over a 2 or 3 year period, and 3 years is a good 3 winters is a good test of Varroa resistance. But this is what happens in a lot of colonies where people get cock a hoop after the first year. They think they've got resistant bees, but at the end of the first year, the mite levels are slightly higher than they were at the start of the year. So, and then it gradually creeps up until you get a problem with the level of vector deformed wing virus. So it gets into sort of dangerous area up here. And that's why you do hear of colonies that don't make the 3rd year. And I think most people who have gone down that route will be familiar with the 3-year blip. So worth bringing up. And then you get Varroa naive bees here. So, that's what's about to happen to Australia. Unfortunately, it's going to be, it's going to be dreadful for them. This second one shows the level of activity that's going on in the dark of the hive. Again, we just did not know this was happening until we did this. This is a piece of brood comb with uncapping, which you're familiar with now at the purple-eyed stage, and you know why that's important. We've put some green around those, and I'm just going to blow up this section in the yellow rectangle here. And the ones marked in blue have all been uncapped and recapped. Now, some of them are really quite obvious. I think that one's quite an obvious one. But when you see it on the frame, you can really see the difference. So back to that. What we did with a heavy heart, and not to recommend it, but just for research, was to take the same picture 24 hours later, just to see what the differences were. Quite a few, huh? So we've marked them on there. So more uncapping in green, a lot of uncapping in 24 hours. One being recapped. It was quite hard to find. If you see another one, let me know. This one here looks like it's been recapped from over there. And then quite a few removed. So a huge amount of activity. And if I can just jog you back to the bar chart of the red and the yellow bars, there was, in England, I think, in the apiary we showed the level of uncapping and recapping was about 60%. Right? And there it is, roughly. There's a huge amount of activity there. And I think that's the point that we can take away. We had no idea it was going on at this level, and it'll also be going on in your hives as well. Right, just going to finish off with just a couple of key points to take away. I think the first one is, one of the things that we can all do in getting towards Varroa-resistant colonies is to stop reproducing from the worst of our stocks. Right? If we do that and we start differentiating, we'll make a huge difference. So this was a colony which wasn't treated, and the best time to see how they're doing is in that spring. And this colony this is a cumulative mite drop rather than daily. Cumulative mite drop, over that time, over 2,000 mites had fallen in those 3 months. And it was in not a very good state, but you compare it to the others that are going on here. So what we would have done in the past, and a lot of people will be doing, will be treating it with formic acid, they'll be doing drone culling, they might be doing a shook swarm to keep that colony going at all costs. And then in due course, if you were successful, you'd be breeding from it. So you'd be replicating those weak traits. And actually what you really need to do is to requeen them. Well, you can requeen in the spring, or you could move it out of area, but that's why we still have Varroa problems after 30 years, because we're in this cycle, the red cycle, kind of recreating these colonies. You want to breed from these. It's obvious, isn't it? Once you start doing a bit of monitoring, it becomes very clear what to do. The other thing is, just to recap, that the beekeeper observations are pretty straightforward. So you need to look for uncapping. Sometimes you can see recapping as well. Just a picture is quite useful in blowing it up, but uncapping is easy to see. You can also see chewed out exoskeleton of pupae, right? And the two together interrupt Varroa reproduction. And in doing that, you create this large cohort of infertile mother mites that gives you low levels of mites. And with low levels of mites, you get low levels of deformed wing virus. And that is Varroa resistance. We have written a book on this. I'm not ashamed to push it a little bit. We have, because we have so we are giving money to Bees for Development, who are in the front here. And so if you do buy the book, you'll be doing some good for charities in Africa, and hopefully investing in learning about Varroa resistance.
I just wanted to ask, in terms of monitoring, looking at mite drops a lot, where you think mite washers fit into the mix in terms of looking at mite levels.
Yep.
And I appreciate that you just have to choose one method and stick to it. Also, just for the female Varroa mites, how long do they have to be without brood before they are infertile?
Let's do the last first, and then you can remind me what the first was. So the point about being infertile is that you've already laid eggs and had a go at it. So they have 2 to 3 attempts at that before they become infertile. And that's why when you're seeing the uncapping, you know that the interruption is taking place. There's some different numbers around that, but it's 2 to 3 is the accepted one. And the first question was...
Go repeat it, please.
Mite washers, so
mite
just
washers.
versus the mite drops,
Yeah.
which, where you sit with that.
No, sorry, thank you. Yeah, no, good question. We've gone down one route, and it's an easy route, so it takes me 30 seconds to look at a board, and I can do it any time, night or day. So it seemed incredibly easy and useful to do. But the real bonus win for us was seeing pupal exoskeleton. And then we started to understand, actually, this is the full process that we're actually able to understand here, which you don't see with the mite wash. Now, mite washes have been done, and again, you just need to be consistent and tune into the levels that they're showing. So if you think about the South African levels, they were doing 100 bees, I think, and they were showing infestation levels. It sounds incredibly accurate, of 10 to 20%. But because it's a percentage, it's highly attractive to people. The reality is it's nothing going to be like that, but it's still you see the consistency. So in South Africa, they went from when Varroa arrived to 10 to 20% down to 4% using samples. And I think similar in Mexico as well. So 4%, around 4% tends to be the natural level if you're using samples.
Normally in nature, when there's a lot of inbreeding in close communities, as happens with Varroa, that results in genetic abnormalities, and there's a drive towards extinction in those situations. And yet with Varroa, which are clearly very inbred, that doesn't happen. Maybe I'm missing something, but do you have any thoughts on that?
Yeah, thank you. It's an interesting question. And I'll just expand the answer a little bit by saying that the haplotype, or the type of Varroa we have, is the Korean haplotype, and there's the Japanese one, which is mainly in Japan, but where it's been found, I think South Africa and Brazil, it gets out-competed with the Korean one. So we all have roughly the same mite, which is quite useful. The time when the brothers and sisters can crossbreed is when there are multiple foundress mites gone in. So there is an opportunity to develop a little bit as a Varroa. So it's not all inbreeding, if you like. So there's a little bit of outside of the family stuff going on, which sounds much more normal, doesn't it? But it's yeah. So they do have the opportunity there. So when you think about how did resistance occur, I'm probably getting into ground I shouldn't do here, but how did resistance occur? Well, there's some of them there which have had the ability to resist those frequently used miticides. Did you want to come back on that? Sorry.
Yes, it's just that the foundress mite is mating with their own offspring. So when is the opportunity for there to be additional DNA introduced?
Yeah, so when 2 foundress mites go in...
Right. Okay, got it.
There'll be 2 sons, 2 sets of daughters.
Got it.
And if there's 3 foundress mites, or 4, can happen as well, then you get the interbreeding families.
Got it.
At that point, though, Steve, won't the colony be close to collapse?
Not necessarily. It can be that there are only a handful of cells left. So it could be that the end, as you go into the winter, or even at the start of in January or late December, when there's just a few cells which have been bred from, all those overwintering winter mites will pile into quite a few. And the result isn't great for the pupa, but it's not great for the Varroa either. They don't successfully reproduce.
You talked about a brood break in the summer. That's how you do it, but you also talked about quite a few others. Is it a mandatory thing in order to run Varroa-free, you've got to have a brood break in the summer. I'm interested in whether that's just what you do as a swarm prevention or whether you're saying that really is a key part of the process.
Okay. So brood breaks are not key to Varroa resistance, okay? They are part and parcel of what bees do. They did it before Varroa and they've done it after Varroa. So they're not part of that whole process. They're helpful, okay? I mean, they help the health of the whole colony, but they're and not something that we try to do either, David. It's just following what the bees do. So we just get a good spring flow where we are, they build up, and then they look to swarm, do a reproductive swarm, and we're quite happy with that. We use that as an opportunity, we'll talk about that tomorrow, to produce a lot of good genetic material for us. So we don't try to have a brood break. The best colony we've ever had for the lowest amount of mites had no swarming that year.
But are you saying, therefore, that you, or at least some of the other people who do it without a summer brood break,
Yeah.
and quite happily?
Absolutely. So it's nothing to do with brood breaks, it's about the level of hygienic behaviour in your bees. It's quite hard to measure, okay, but it's, when you start seeing these activities, the uncapping and the chewing out, you start to get a feel for it. But the brood breaks are interesting, they're slightly irrelevant, okay? And so people talk about multiple brood breaks and small colonies. That's just irrelevant to us. People, what they're doing is extrapolating from very small examples.
You were saying you were open mating your queens.
Yep.
So you might think that hygienic behaviour, if it's genetically controlled, would get diluted fairly quickly. Do you think your success is because you're also saturating the area with drones that are also carrying the trait?
Yeah. Not to start with, no. You think about how many treatment-free beekeepers who haven't treated for 8 years were on that map. They've all started from somewhere, and they won't necessarily, when they've started, they won't have drone saturation at all. So what we see is really strong heritability of the Varroa-resistant traits down the queen line. That's what the USDA found as well, and there was a recent experiment done with a queen swap with Isabel Grindrod and Stephen Martin, where they put the resistant queen in with mite naive bees from colonies, and then they put the naive queen in with the resistant bees, and the resistant queen made that colony resistant. So, but in the mating, we find it follows. Okay, so do you remember the map, the full year when the daughter started laying? She had the same traits, and we found this quite early. So we started trying to understand why it was, and we found that the USDA had found exactly the same. So that's the really good news, is it follows down the queen line. Now, the other thing I would say, again, just an observation, is that in the first 3 years or so, before you do have your drones out there, the traits are a little bit more volatile. But now we have almost complete stability, but a high level of stability in Varroa resistance, and that is partly with your drones flying. So we sort of think 3 years to get established and 5 years to have all your drones out there as well. So we do quite a bit of work with our drones. They're absolute heroes, and we've found some new roles for them, which we'll talk about tomorrow.
You had on your map the Loon Valley Association, 70 colonies there.
Yeah.
They have an awful lot of long hives, horizontal hives.
Yeah.
Do you think that might, where they don't intervene during the season either?
Yeah.
What do you think about, could you comment on that?
Yeah. So we were at a conference run by Loon Valley at the weekend.
Oh, really?
And the Loon Valley, they have their own hive, okay, which is the size of a chicken coop, and they use all natural comb in it. You know, no supports or anything. So it's an interesting thing. And when you don't intervene and you don't look at brood comb as much as us lot, you actually attribute all sorts of things to Varroa resistance. And I think a lot of natural beekeepers are saying. So the less you interfere, and some hives perhaps are a little bit more bee-friendly than others, the more chance you'll have of a healthy colony, let's put it that way. But if you put mite-naive bees in a Loon Valley hive, forget it, they wouldn't last, okay? So what happens is that because people don't look at the brood comb very much, they attribute some of Varroa resistance to other things, which we had the sort of satellite things, which are the ancillary helpful environmental benefits. But actually the key is in that worker brood, okay? That's where the sex and violence needs to happen.
Do you know anything about the importation of bees? What effect that is having or not having?
Yeah, so we don't go anywhere near imported bees, okay, if we can avoid, and partly because we do not want to upset or bring in new genetics into the area. So we already have genetic gold, I think, which is locally adapted bees, adapted to our microclimate, adapted to our pollen availability, our nectar flows, probably our local pathogens as well. And you're not going to improve on that, we don't think. You might better boost it artificially for a year or two, but then you're on the phone after another queen. So we try and avoid it. Now, my understanding is some of the bee breeders tried to copy what the USDA did a few years back now, and I don't think it's been that successful. Right? So, but we haven't tried them, is the honest answer. So we can't be sure, but we would not want to. And they're very narrowly bred, okay? So they're bred for sale, which I can see the use for them in commercial situations, but we don't think they're bred for fitness, and that's what we want. We want bees that we can stay with, which we can work with, improve, and that seems to have worked incredibly well. So I can't see anything being better than that for the short term. The problem that the bee breeders have got is the genetics of Varroa-resistance is incredibly complicated. Now, we haven't nailed it at all, and we're nowhere near nailing it. You know, the best funded project at the USDA didn't get there, and they're looking for genetic markers that they can just breed from and dish out. They came out with something called pole lines, which is not quite there, but it's not bad. There was a bit of work out in the spring from France, which showed that the Varroa-resistant traits, they're called polygenic, so they're additive rather than dominant or recessive. And that's the latest work, and it's the improvements of being able to understand the whole honeybee genome is allowed them to do this. But the Varroa-resistant traits came from 60 different parts of the genome. So you try breeding from that. And we're going to find out more, okay? It's very complicated. It's very complicated. The bees have sorted it. All we need to do as beekeepers is to look at the expressions of those traits.
On your graph, you said there was very little hygienic behaviour with the drone brood. Would that mean that it might help if you took frames of drone brood out and killed it in the spring to kill the Varroa and reduce the overall load going into the early summer?
Okay, thank you for the question. I obviously haven't explained that very well. So the more we take out, whether it's through drone culling, whether it's from treatments or queen frame treatment, the easier time that the bees will have in sorting out the Varroa, and the less hygienic traits we'll be able to assess. So we don't do that, and to us that's part of old thinking, okay? So our thinking is that the drones are the absolute heroes in the spring, because they take the mass of mites, which otherwise would be in the worker drone, and the attraction is the amount of chemical cues coming off the larger amounts of drone larvae. So they're attracted to drones, and so those mites are going from workers to drones, which allows the workers to be out foraging and building up ready to swarm. So the drones take the hit, and then there's a swarm, and it moves on from there. And the natural selection pivot is that the bees which have less mites than them will have less, because of their hygienic behaviour from January to March, will have less mites in the drones. And those are the drones which have more chance of successfully mating. So that's a really important natural selection point. So we don't try and do that, otherwise we're just going to kid ourselves that things are great. And actually then we might breed from the wrong bees as well.
Leading on from the last question a little bit, you mentioned at the beginning of your talk that Apis cerana, the mites are concentrated in the drones. In fact, they're all in the drones and not in the worker bees. Why is that? Because I've done reading and been told that there happens to be more in drone brood anyway than there is in worker brood. Is it a size thing, or is it a chemical signal thing, like you say? What happens in Apis cerana that causes that? I
don't know. There's some of the work has not been that interesting. Well, has been interesting, but it's been inconclusive. And some of it is about the worker bees don't tolerate any Varroa in there. And and we'll remove it. And the other thought is there's some chemical release which doesn't allow them to successfully reproduce in worker brood. But it's not something we've looked at very closely, I'm afraid.
You mentioned briefly earlier on the possibility of, I think you were talking about America, saying that people were looking to sell what they call Varroa-resistant bees.
Yeah.
I'm just wondering, you're talking about so much of the queen line being the important thing and the heritability.
Yeah.
If there's any sense in which there's any kind of possibility of queen sharing or breeding for other divisions, if working with other divisions who might
Yes.
be doing that.
Yes. Yes.
Oh, okay.
We'll talk about that tomorrow. We have, Steve's here today, in due course, okay, we've just set up, we're a bit of a federal model. We've worked together with some of our members here who started off, and we sort of share between us and share with others. We've just set up a queen rearing apiary, which has got some of our best resistant traits in it, and someone who's incredibly enthusiastic to get it going. It will take a while, okay, but we're very happy to try and help out, particularly where we've made friends with Chichester, and there's lots of other clubs who are sort of on their way, you know, and starting projects. So I'm sorry to hear you're not quite there yet, but there was great engagement. And I think, you know, I would just say to everyone, just take your time, okay? There's lots of mite susceptible bees out there because we've been breeding them for 30 years, all right? So just spend your time monitoring and looking for it. You should be able to find it, I think. If not, I'll come down and have a look.
Thank you very much.
Okay.
Thanks, Steve.
Just a quick question. Do you have a view on bee gyms?
I think what we're trying to do, let me answer it in a slightly different way, is to understand what bees are naturally doing in the wild. So the research on why bees survived in South Africa or in Cuba, how have they done it, you know, and then try not to make up something clever and man-made. Let's just copy what they do. And to us, that's long-term and sustainable, permanent solutions. So the bee gym, we've had no experience of at all. From our perspective, all of that graph we showed, which shows the amount of worker brood through the year versus the amount of drone brood, is the key to it. And it's just that those bees from those Varroa from Asia just never had so many opportunities to reproduce, and reproducing in worker brood is their thing now over here, and our bees stopping them from doing that is the point of resistance. Can a bee gym work? I don't know. I mean, it's, you know, the activity is in the brood nest, really, I would think, but there's a lot of people who do believe in them, and particularly those who are selling them, but we've no we've no experience, so apologies. Yeah.
Early on in your talk, you said how the Varroa mite doesn't like a particularly warm hive. So does that mean that the level of insulation we put in, say, on the winter, might be a good idea to keep it going right through the whole year?
Yeah.
And also, are polyhives therefore better?
Yeah. Did I say that? We use insulation, okay, all year round. And for summer, just to stop the temperatures. So we put these bees in thin-walled wooden nationals, you know, for not a good reason, not for the bees' help. So we have insulation for the summer, and then we have more insulation for the winter, just recognizing actually the winter is the time where the greatest weather stress is going to take place, and and there's going to be a smaller colony at that point in time. We don't buy into it's all about heat and it's all about humidity. Okay, we're just taking the stress of managing the temperature changes away from them as best we can.